عليه اختبارات.
انا اختبارات ولا شيء انا سويت.
قال لي بس هذا يونت 7 بديت استنى يونت 7 القاعده اللي فيه شوي خربط.
ليش؟ عليت الكوزات. انا وصلت
يونت 7 نتيت ما اخذناها.
وصلت للثالث انا بس.
نسيت الموت ويانا.
زين كيف تحلهم وما اخذناهم ويا عمر شريف؟
سهلين كلها حتى الفكاب سهل. لا
قلت كل ما نخلص
متوسط. يعني في الخيارات ما في شيء متشابه. يحط لك المعنى ذا والباقي
كلهم فادي.
لا مو على كيف قالوا لا اذا يمدي في الخطه يخلصون 200 الكويز زين شلون نحلها؟ طلعت لك حاجه خش يلا كم تربع تعشين
يمكن يسوون علمت الادب.
Okay, before we start, I'm just going to run this video on the structure of the DNA and chromosomes. We didn't get time last time to run it, so we'll just run that one so you can have a look at it. So this covers lesson 1.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. The nucleosomes are packaged into a thread known as chromatin. This fiber is then looped and further packaged using other proteins which are not shown here. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. Chromosomes are not always present. They form around the time cells divide when the 2 copies of the cell's DNA need to be separated. At other times, as we can see now after the cell has divided, our DNA is less highly organized. It is still wrapped up around the histones, but not coiled in the chromosomes.
So, in lesson 2, we talked about the cell cycle and how the cell cycle has interphase and the mitotic phase. So in interphase, we said that the cell is rapidly growing. So it's between the last division and the next division. And this is where the cell duplicates its organelles. It prepares for the next cell division. So that's the interphase. It's the longest period of the cell cycle. So then the second phase is the mitotic phase, which is the actual nuclear and cytoplasmic division. And this is what we'll talk about in this lesson today. So the mitotic phase, it's separated into 2 different stages. The first is the what we call mitosis. This is the nuclear division, where the nucleus divides. So the chromosomes divide to the 2 new daughter cells. So remember, in the S phase, what did we do?
Replication.
We did replication, we duplicated the DNA. So that DNA is now separated in the mitosis, in the nuclear division, into 2 new daughter cells, okay? So we have the parent cell. When it divides into 2, the new cells that arise from that parent cell, we call them daughter cells, okay? So once nuclear division happens, then you need to separate the two cells. So the second step is cytokinesis. Basically, is referring to the division of the cytoplasm.
Okay.
So the two new cells will have equal cytoplasm, and they'll have one nuclei each when after cytokinesis.
Okay.
So this forms the two new daughter cells.
You have the nuclei, not the nucleus.
So yeah, each one will have a nuclei. Yeah.
Then it will become nucleus.
Well, yeah. It's nuclei is plural, nucleus is single.
Oh.
Okay. All right. So the actual mitosis stage is divided into 4 separate phases. Okay. So we have this initial phase is the prophase. This is where the chromosomes start to appear. Okay. And the nuclear membrane disappears in this one. The nuclear membrane disappears, the cytoplasm, the chromosomes start to appear, the nucleolus disappears, and what happens is the centrioles, remember we talked about the centrioles? Yeah. They're important in this structure called the spindle, mitotic spindle. So it forms this mitotic spindle. It's formed from microtubules, okay?
Fibers.
Microtubule fibers, okay? And these are important in the separation of the chromosomes. So you see here the chromosomes are they unduplicated or duplicated?
Unduplicated.
Duplicated, remember?
Okay.
Because we've already passed through interphase. The chromosomes have been duplicated, okay? So the S phase is where you get duplication of the DNA. So you see, if you have a look at here, we got each duplicated chromosome has 2 sister chromatids, remember? Attached at the centromere. Yeah. So that is a duplicated chromosome. And this is where that centromere is very important, because the centromere is the point of attachment for the mitotic spindle, okay? So the microtubules attach to the centromere, and then this is where they pull and separate each chromosome to the new daughter cell, okay? So in prophase, the chromosomes start to appear, so they condense, okay? The chromatin condenses into chromosomes, they're more visible, and then the nuclear membrane disappears. Obviously, it has to dissolve because the chromosomes need to be separated. The nucleolus disappears. The centrioles, they migrate. They move to each pole. Remember, the Earth has 2 poles, north and south?
Yeah.
So on each side, one centriole will be, okay? So it forms the mitotic spindle to pull the chromosome to each side of the pole. How this works is that, remember microtubules, they're made up of tubulin?
Yes.
If you have a chain, you know what a chain is? This a metal chain?
Yeah.
It's got little, I don't know what you call them, rings, okay?
Okay.
So if you pull rings out of it, you make it shorter, correct?
Yes.
If you put more, you can make it longer.
Yeah.
The same thing happens here. You can shorten the length or make it longer, the spindle fibers, by adding or removing tubulin to the fiber. So basically, if you want to pull the chromosomes, what you do is at this end, you just keep removing the tubulin molecules, and it pulls it, okay? And it requires ATP, so it is a energy dependent process, okay? So the spindle fibers begin to form, or as mitotic spindles. The centrioles are moving to each end of the pole, and then we move on to the next stage, where we call the metaphase, okay? So in this phase, remember how many chromosomes we have?
We have
23 pairs, so 46 in total, okay?
Yeah.
So in mitosis, and I want you to know this because there's a difference between mitosis and meiosis in this particular arrangement. In mitosis, you get all 46 lining up here, okay? So remember, 46, they all line up. So we get 46 when it separates the duplicated chromosomes, you get 46 here, 46 in the new cell. Okay? Do you understand that?
Yes.
Because it's duplicated. Okay?
Can you repeat the last sentence?
So you have 46 duplicated chromosomes. Okay?
Yeah.
So they're 46 duplicated. After cell division, you end up with 46 unduplicated in each cell. Okay?
Okay.
Is that clear?
Yes.
So it's duplicated here in that form. So when the 46 separate into unduplicated, you end up with 46. And this process will continue again. You will get DNA replication, so the chromosomes become, after DNA replication, duplicated again, correct? Okay. So in metaphase, what's happening is that the chromosomes line up at the equator, okay, or at the metaphase plate, another name for it. So at the metaphase, so right at the center, they line up, and they're attached to the mitotic spindles at the centromere, each chromosome, okay? Got that?
Yes.
So that's metaphase when they line up. So this is an actual image of a cell in metaphase. This is an image in prophase, okay? Then what happens is, as I said, tubulin monomers are removed from the spindle fiber, and this requires ATP, and the mitotic spindles shorten. As they shorten, they pull the chromosomes apart, and the centromeres start to dissolve, okay? So allowing for the separation of the chromosomes, okay? So in anaphase, okay, the chromosomes start to separate, okay? The mitotic spindle is shortening and pulling the unduplicated chromosomes to each side, to each pole, okay? So these are known as the poles. Like we have, you know, the north and south pole on the Earth. So once the chromosomes arrive at each pole, then you get the next step, which is telophase and cytokinesis, okay? So in telophase, what's happening is the reverse of prophase, okay? So the chromosomes are starting to decondense, the spindle fibers are disappearing, the nuclear membrane is appearing again, the nucleolus is appearing again, and in late telophase, you're getting this bulging, okay, of the cell, which we call the cleavage furrow, okay? So this is happening because of microfilaments, okay? Remember microfilaments?
Yes.
They're made up of actin.
Yes.
So the microfilaments actually form a ring around at the middle of the cell, okay? So it can separate it to equal two new cells, all right? So this ring of actin, it constricts. You know what a constriction is?
Yes.
So, you know, what should I say, you got you hold something and you squeeze it, okay? So squeezing, constricting it, squeezing the cell at the center. You see, and this cleavage furrow, this thing forms. And this constriction continues until in cytokinesis, you get separation of the two cells. So basically, you put a string around this and pull it, into a knot, and what happens?
It
cuts it into two. So this cleavage furrow is formed by actin filaments that are making this constrictive ring that is pulling and eventually separating the two new cells into two new daughter cells. Now I wanted to explain that using the image because it's simpler to show it. There's a lot of text now that explains that same thing I just talked to you about, and we'll go through that, okay?
Doctor, what's happening in the prophase?
So prophase, the nuclear membrane is disappearing, the chromosomes are starting to form, So a bit visible, the chromatin is condensing into chromosomes. The spindle fibers are starting to form. The centrioles start to migrate to the poles, and then what happens in they start to attach to centromeres, and then in metaphase the chromosomes are lined at the equator or at the metaphase plate at the center, ready to separate them into two. One other thing to notice is that you see this is a duplicated chromosome, huh? So it doesn't matter if it lines up this way or this way, the same copy will end up in each cell. This is very important because in meiosis, we end up with different, we get crossing over, which creates non-identical sister chromatids, okay? Here they're identical. In meiosis, we get non-identical because it's very important for diversity. You know, that's why you don't look like your brother or your mother exactly.
Yeah.
Because you don't have the exact same genes, okay? They've shuffled, okay? Crossing over and other steps, okay? Huh?
They're like
Independent assortment, crossing over, these mechanisms, okay? They help to give diversity. So here, it doesn't matter if the chromosome lines up this way or this way. It's gonna end up the same. If in meiosis, this chromosomes, because they're different, if it lines up this way and the next one lines up that way, that means this cell will not get the exact thing as that. They will get
Another thing.
different version. That's why each sperm is unique, each egg is unique. We'll talk about when we get to that later. I just wanted to introduce that because it's some students don't understand that sometimes, but you will understand it when we compare it to mitosis, okay? So, as I mentioned,
but Doctor, the metaphase is still duplicated, no?
in the metaphase, yes, the chromosomes are duplicated.
Mitosis will كلكم duplicated.
They become unduplicated once they in anaphase. Okay, so as I mentioned before, the cells that are formed from mitosis are identical to the original cell, okay? They're identical to the original cell because you got DNA replication, and as I mentioned, there's no crossing over, there's no independent assortment or so on. So you're getting exact copies. So the cell that you came from has the same DNA that you have now, okay? So all your cells will have the same DNA as the original, except for the gametes, okay? They're going to be different, obviously. So what mitosis is, as I mentioned, is you get nuclear division and cytoplasmic division. So the mitotic phase, okay? So the cytoplasmic division is cytokinesis, the actual separation of the cytoplasm. So you got the two sister chromatids. Remember the two chromosomes?
yes.
Sister chromatids, they're duplicated, okay? So they're duplicated and they separate. So the sister chromatids which are duplicated separate during mitosis. And it always happens in diploid cells, okay? So mitosis happens in diploid cells. So 2N. And it produces diploid, okay?
Yeah.
So the end result is diploid. As I mentioned, remember, I said you got 46 duplicated chromosomes and they separate into 46 unduplicated, okay? So you started off with 46, you end up with 46. So you start off with 2N, you end up with 2N. Because the 46 chromosomes we're talking about is 23 pairs, okay, of duplicated chromosomes. Okay? So you start off with 2N and then the resulting cells, the daughter cells, are also 2N. So in humans, we got, as I mentioned here, 46. They become duplicated, okay? During replication, they go mitosis and cytokinesis, and each cell now will have 46. Any questions on that? Does he like to sleep in the classroom all the time? Sleep time is at home. Okay. You're not going to learn anything if you sleep in the classroom. You might as well be at home. Okay. Yes. So, because of DNA replication in the S phase, okay? Because of DNA replication in S phase, the cells are genetically identical to each other. The daughter cells are genetically identical to the mother cell. So you have the mother cell.
The same for the daughter.
So they all have the same. All these three cells are genetically identical. Okay. In fact, all your cells in your body, the trillions of cells, are genetically identical. There might be slightly differences, through mutation, but they are genetically identical. Okay? This happens. This allows for complex organisms like us to have the same set of chromosomes in each cell. So the trillions of cells that have arisen from the single zygote have the same chromosomes, exact same chromosomes. So mitosis ensures that all your cells have the same chromosomes that you started off with. And remember, before I told you that mitosis is very important for different processes. So growth. So you're starting off from one cell and then you're growing into trillions of cells, okay? So the process of mitosis is important for that. Cell replacement. Your blood cells, your skin cells, your epithelia, many of the cells, immune cells, they're replaced all the time. So how do you get new cells? Obviously, you have to have mitosis, cell division. So mitosis replaces cells. Regeneration, okay? So if you cut yourself, and you have an injury, tissue is repaired by mitosis. Cells replicate, okay? So you get regeneration of the tissue. Some organisms, they reproduce by this method. So in asexual reproduction, some single cell organisms, they just split into two. And that's it. Bacteria or some protists, okay? And as I mentioned, the 4 processes or phases of mitosis include the prophase, the metaphase, anaphase, and telophase, okay?
Telophase is not cytokinesis?
No. Telophase, cytokinesis is, so you got the mitotic phase, which includes mitosis and cytokinesis. So mitosis includes these 4 phases, then you have the cytokinesis, okay?
The cytokinesis is the division of cytoplasm.
Yeah, so this is the division of the nucleus. This is the telophase is a part of the mitosis, okay?
But what will actually happen?
So at the end of telophase, you start to get this cleavage fera, okay? But it's not the complete. So when you get
The
complete
cytokinesis is
separation
complete.
of the cytoplasm, this is cytokinesis, okay?
This is in plants, doctor?
Sorry?
The cleavage for it's in plants or animals?
Both.
Both?
Yeah. In plants, what you get is that you have a cell wall, okay? The cell wall doesn't actually separate. What happens is this membrane separates into two, and then eventually you get formation of a cell wall between them. Okay.
Oh, the cell wall.
Doctor, and the mitosis and cytokinesis, it's all mitotic.
Yes, mitotic. The mitotic phase. So the mitotic phase is mitosis and cytokinesis. All right, so I'm just gonna go through this quickly because I already been through it just in the picture. So as I mentioned, the chromosomes start to become visible. So the duplicated chromosomes. When did they duplicate again? When did they duplicate?
Prophase. Anaphase. Anaphase.
Prophase.
The S.
S phase. Come on, guys, wake up.
Prophase and S phase, both.
S phase. They duplicated. Okay. DNA replication happened when?
S phase.
S phase. Okay. So the duplicated chromosomes become visible. So therefore, before they're in the form of chromatin. Now, when the cell is starting to divide, actually, you're getting the formation of the chromosomes, because these chromosomes, as I mentioned before, they're nice little units that can be easily separated rather than chromatin all over the place. Okay, so it makes it more efficient to separate the genetic material into the two new daughter cells. Okay, so the chromosomes start to appear. The mitotic spindle between the centrioles form, so you got remember the centrosome? You got the pair of centrioles. Those pair of centrioles then start to separate into each pole, okay? So remember the poles of the cell?
Yep.
So you get separation of the centrioles, formation of the mitotic spindle, that microtubule fibers. So, they move to the different poles. You get the nuclear membrane dissolving, the nucleolus disappears, okay? So this gives now access for the cell to the chromosomes to separate them. So it's common sense to remove the nuclear membrane, okay? So we get the centromeres attaching to microtubules on the opposite side of the cell, and this is where metabolic activity decreases, okay? Because mostly it's concentrating on actually getting this cell to divide now, okay? So the concentration of cell division, not on other metabolic activity of the cell.
Doctor, what's metabolic activity?
Normal cellular processes, okay? Metabolism.
Oh, okay.
Okay? It's more concentrated now on dividing the cell rather than doing other activities, producing other molecules and,
Just separating.
it's got more other priorities at the moment.
And it's involved in division, metabolic?
Well, so, the cell has normal metabolic activity, what it's doing, what its function is, okay? Every cell has different functions, okay? So, whenever its normal metabolic activity is, at this time, it's decreased because it's more concentrated on trying to get these other processes done. I don't know the metabolic activity of each different type of cell.
Yeah.
All right, so in metaphase, we said that the duplicated chromosomes, they line up at the center, the equator, okay? So this one plane, okay? And as I mentioned, this is also called the metaphase plate, okay? So this center. Now, so there's some tension put onto the chromosomes. They're starting to pull them, okay? But they're not separated, okay? So they line up in the center. There is some tension on the chromosomes, but they don't separate yet, okay? Because the separation happens in the anaphase, okay? So the sister chromatids don't separate. There is some equal force on each of them. So yes. So the sister chromatids, okay? The two sister chromatids are actually duplicated chromosomes, okay? So here they're also, now referring to them as daughter chromosomes, because once they end up into each of the new daughter cells, they're going to be daughter chromosomes, okay? So they pull towards the pole by these microtubules, and remember the tubulin, monomers that make up the microtubules.
Again, the one?
The tubulin, okay? So remember from chapter 3, microtubules is made up of tubulin. So as you remove these, what is it happening? It's getting shorter and shorter. So here, there's actually the fibers are getting shorter because the monomers are being removed from it. And one other thing I didn't mention is that the microtubules actually come from the cytoskeleton, okay? During cell division, the cytoskeleton disassembles, okay? And the tubulin is used in the spindle fibers, okay? From the microtubules in the cytoskeleton. So during cell division, the cytoskeleton also disassembles. This is not in here, but it does. So these, you would think where this tubulin is coming from. The cell needs a lot of it for cell division. It's actually coming from the cytoskeleton. Okay.
And it dissolves with the.
So the cytoskeleton disappears as well at the same time as the nuclear membrane. So those tubulin monomers are released, and they're used to form this mitotic spindle. And this action of pulling requires energy. Okay. So they're acting like motor proteins. Okay. So there's actually movement going on. So they're motor proteins. Okay, involved in this. So each one of those centrioles is pulling to opposite ends, okay? So this one is pulling this way, that one is pulling this way, and you get separation of the sister chromatids, okay? So by the time this process is finished, you will get an exact copy of the 46 chromosomes on this side of the pole and an exact on the other side, okay? Ready for cytokinesis, okay? So once you get this, you know, it's the beginning of cytokinesis, okay? In telophase, you get the beginning of cytokinesis. It's not complete, okay?
What's motor protein?
Motor proteins, like proteins that help to
move,
for movement, okay? Like the flagella, okay? Like the cilia, they're motor proteins, okay? The same thing here. There's obviously movement happening there.
Yeah.
The chromosomes are being pulled, so this is a motor action. It requires motor proteins. So we go into telophase. Now in telophase, it's very easy to remember because it's actually the opposite of prophase. Okay? It's the reverse, okay, of prophase. The reverse is happening, you know. What you've got the chromosomes arrived, okay? They start to decondense. The nuclear membrane starts to form around them again, okay? So, and the mitotic spindles, they start to disappear. Remember, they appeared in the anaphase. Now they're disappearing, okay? So the mitotic spindles disappear. Nuclear membranes reforming, and the chromosomes are now uncoiling and forming chromatin again, okay? Ready for interphase again, okay?
They're forming chromatin.
Chromatin, okay? They're starting to decondense into chromatin.
So we call it unduplicated chromosome.
So, well, we call it unduplicated chromosomes, but they're going to be there for only a short period. We don't need to, remember, during normal cell activity, you don't want the chromosomes. They compact, condense. The cell needs access to the genetic material to make proteins, to make RNA, okay?
So they come back.
They need to decondense, okay?
It's
It's only for a short period. And that's why, you know, as I said, the actual division of the cell is only a short period. Most of the time it's spent in interphase. So this happens very rapidly. So, I mean, they exist, but then they disappear. So the unduplicated chromosomes exist after anaphase, but then they disappear. Because, as I said, chromosomes are only needed during the nuclear division. After that, their function is over. It makes it easy for the cell to separate the genetic material, because chromosomes are nice little units that can be separated. Chromatin, on the other hand, is not. Yeah.
But Doctor, now when we separation the chromosome,
Yeah.
we have just one, and it will be duplicated when we.
So in the
after
next
the
cycle,
division.
remember, you got mitosis, all right, and then you have the interphase, okay? So the mitotic, and you have the interphase. So here, you know, is the cell division. Then you have the G1, then you have S, then you have G2. So the DNA, once you get the cell division, then it comes into this phase again. DNA replicates again, in S phase. So here it will become duplicated again.
Yeah.
But in the S phase, it is chromatin.
Yeah, it's chromatin.
So what happens in the G1 phase?
G1 phase is growth, duplication of the, because it's just separated into two, so it's now need to grow again, the cell, and duplicate the organelles, okay? Increased metabolic activity, increased growth, duplication of all the organelles, okay?
So there's duplication. So
Yeah.
duplication happens in the G1 and the S phase.
Yeah.
And doctor, there are checkpoints.
Duplication of DNA.
But DNA is S phase.
Here is not DNA, I said organelles,
Organelles. Yeah.
okay? Mitochondria,
Okay, yeah.
you know.
Doctor, there are checkpoints at the end of any
Yeah,
process?
we are not going to take this, but there is checkpoints to make sure that these processes happen accurately. So at the end of DNA synthesis, to make sure that the cell has correctly duplicated the
In every
DNA.
phase.
Yeah. So well, in certain phases, there is checkpoints.
In G phase, S phase,
Yeah.
G2 phase.
So the book also talks about these, but I think we're not going to cover them. We don't have time.
Okay. If there are any wrong.
If there's a mistake, the cell will go and correct it, okay? If it can't correct it, it terminates the cell. It goes into apoptosis. And in cancer, some of the
What's
checkpoint proteins in cancer have become malfunction. Like
That's what-
P53 is one of the major ones, okay? And so the cell cycle proteins, okay? There's very important cell cycle proteins that keep a check on each of these stages. Yes.
Are these proteins that next to checkpoints are the ones that she talked in the video? She says that there is a proteins, but they are not shown in the video.
No, that's different. The checkpoints are different, okay? So checkpoints, they're different. They're important in making sure that the cell is prepared for the next phase, okay? If it's not, it'll try to correct it. If it can't, it causes the cell to die. Because you don't want a non-viable cell in your body. But sometimes it fails, these checkpoints, because there's mutations in the genes for these checkpoint proteins, and you get cancer, where the cell can just go without nothing is going to check it. It's just going to go.
So if it didn't die, it become cancer.
Yes.
Okay,
Well,
so
not always. Some.
this is the main
Depending
reason for cancer.
Yeah, main reason is these checkpoint proteins.
And
One of the main one is P53, I think. P52 or 53, I forgot.
Okay, but if the receptor is not working, and
What
they
do you
are
mean
just
receptor?
produced when maybe like pancreas, when you need to synthesize insulin.
No, nothing like that. That's different. In
There are
diabetes,
instructions come with
you get destruction of the beta cells. The immune system is killing them.
Ah.
It's a different thing.
So tumors are made if there are no checkpoints.
Yeah. So there is other mechanisms that cancer use. They can mutate in proteins which are important for growth. Survival. Sometimes some proteins are mutated that inhibit apoptosis. Okay, so it prevents the cell from dying.
Yeah.
Keeps it alive. Okay, makes it grow faster. Sometimes they release growth factors to make more blood vessels so they can grow faster. All sorts of things. Cancer is not just one disease. It's a multiple diseases, and it's very complex. There's a lot of research going on it.
Doctor, you have said that in mitosis we start with 2N and we end with 2N.
Yeah.
But in the telophase there's only one strand chromosome, one chromatin. Why is that?
2N, okay?
Yeah.
46. You get S5. What did you end up with?
Replication.
Duplicated.
So you get a 2N, 46 duplicate, huh?
Yeah.
This is unduplicate. So each chromosome is now duplicated, okay? All of the 46.
Okay.
When you separate them, you still end up with 46
Unduplicated.
unduplicated. Now, even though these 46 are duplicate, they're identical. So there's still 2N, okay?
Doctor, I didn't get it.
Doctor, so it means that in the teal phase it ends with 46 and 46.
Yeah. So time. How many slides do we have? One. Okay. I'll concentrate a little bit more on this. So, let me just write it here. It might be easier. Okay. So you have 46 chromosomes. Then you have the
S phase.
S, okay, S synthesis phase, okay. What do you end up with? So this is unduplicated, correct?
Yes.
Yes.
You got 46.
Duplicate.
So this is 2n. This is still 2n because it's just 46 duplicated, okay? So in the mitosis, what happens?
Separate.
You get the separation of the duplicated chromosomes, okay?
So it becomes
So-
46 and duplicate.
You get separation of the duplicated chromosomes, okay? So you have 46 here duplicated. When they separate, you get 46 here. So this is not showing all of them, okay?
Unduplicated.
And in 46 here, unduplicated on each side. So it's still 2n. This is 2n as well. You got that? Still sleeping? Okay. Do you have exams? Okay, studying for chemistry. Is chemistry hard?
No, so easy.
Easy?
No.
Easier than biology?
Yes.
Easier than the Islamic.
By the way, are you guys happy with the exam results or not?
Wallah, we're not happy.
You're not happy? I
He
saw the exam results.
He got 15 and he's not
This
happy.
guy, Muhammad
Abdullah.
والله
Yeah.
He's a genius.
يا ركز شوي معك. انا توقعت انه 15. بس
Okay.
كم اخذت؟
15.
14 صدر 15.
قاعد.
Okay.
والله زعلان.
All right.
جايكم من الرياض يا عيال ذاكره.
So as we said, telophase is the reverse of prophase. Everything that happened in prophase is now reversing, and I mentioned all of those. And it's the beginning of cytokinesis as well here. So this cleavage phara, as I mentioned before, starts to form, constricting the cell.
Yes.
Okay. So if you have a look at the cleavage phara here, this is the cell undergoing cytokinesis. You know, you get you're making bread at home, your mom, and she's got the dough and everything, and she pinches off a piece. Exactly, you know, you're squeezing, pulling a piece off. This is what's happening here. You're just pulling it into two equal cells, okay? So, and the important thing is, in cytokinesis, the cytoplasm is equally distributed. So you got the microfilaments, okay? So, okay, the actin, okay? Remember?
Yeah.
So it coils around it at the middle of the cell and starts to tighten and constrict the cell, forming this structure. So this is called a cleavage phara right here. This bulge in the cell. You know what a bulge is?
Yeah.
So the cell is bulging, you get this is called a cleavage phara. And this is eventually what will cause the cell to separate into two, as it's constricted by the actin filaments.
Doctor, maybe please repeat just what you said.
Huh?
Maybe please repeat what is cleavage forums?
So you're getting cytokinesis is cytoplasmic division.
Yeah.
Microfilament proteins, okay?
Okay.
The actin is important in that. The actin forms this constrictive ring around it, okay? And tightens, just like, you know, you put a rope around it and put a knot in it and pull, okay? So it's tightening at this midsection, okay? And as it tightens, this bulge forms, okay? This bulge here, you see that? This is known as the cleavage phara. And this continues to bulge. This is known as this this microfilaments, okay, this actin, they form this what is called a contractile ring. It means it's contracting. Okay?
Oh, so
This ring, so like this actin ring is known as the contractile ring. So as it's pulling, okay, tightening,
yeah.
it forms the cleavage furrow, and eventually it'll tighten so much that it'll pinch the cell into two new daughter cells, okay? So it'll pinch it off. This is what it means, pinch. You know how you're making dough and you pinch a piece out of it? The same thing, you're pinching it off. So once that happens, you get the disassembly of the microtubules because they're no microfilaments, sorry, they're no longer needed. And at the end, you have two identical daughter cells because we had the duplication of the chromosomes. Remember, the sister chromatids are identical. So when they separate into the two new cells, you have identical two new donor cells formed from the original cells.
Doctor, basically for the active filaments, they at the midsection they pull.
They pull just like a, you know, you put a rope around it and pull, tighten it. Okay, as you tighten it, it gets smaller and smaller and smaller. Eventually, it'll break off the two pieces.
Small, then pinches off.
Yeah.
Okay. And it's like the microtubules will disappear at the end.
Yeah, it will disappear obviously. You don't need it. Okay. Disassembles, alright. The microtubules, yeah, same sort of thing. So the end result in mitosis is two identical cells, and they're diploid. Okay. The other thing to remember, diploid, 2N. 2N. 46 unduplicated. Unduplicated, right?
For 46 and the unduplicated chromosome in each cell.
All right. We got some time. Let's watch this movie on the cell cycle. 3 minutes, huh?
Human contains an estimated 100 trillion cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. In eukaryotic cells, this process is accomplished by a series of well-orchestrated steps called mitosis. Every day, our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material. Prior to cell division, where the cell divides into two identical cells, the DNA needs to be replicated so that each daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes. This beads on a string structure is called chromatin. As a cell prepares to divide, chromatin coils up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into 3 distinct phases. During G1 or Gap1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S or synthesis phase, the DNA replicates. Finally, during G2 or Gap2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase, and a very short period of time actually dividing, a process called mitosis. The cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, appearing as 2 sister chromatids held together at the centromere. The cytoskeleton disassembles as the spindle begins to form. In animal cells, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelope breaks down. Towards the end of prophase, chromosomes attach by proteins in their centromeres called kinetochores to microtubules from each pole, moving the chromosomes toward the equator of the cell. During metaphase, all chromosomes are aligned at the equator of the cell, called the metaphase plate. Anaphase begins with the degradation of proteins that hold sister chromatids together, freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filaments surrounding the inside of the cell's circumference. Chromosomes cluster at opposite poles and begin decondensing as the nuclear envelope reforms around them. The spindle apparatus disassembles as the microtubules are broken down into tubulin monomers that can be used to form the cytoskeleton of the daughter cells. In animal cells, cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the two identical daughter cells. During the cell cycle, certain checkpoints are encountered to make sure the process is occurring accurately. And if it is not, the cell cycle will stop at the checkpoint and correct, or possibly inhibit that cell from dividing. The first checkpoint is the G1S checkpoint, and is considered the primary point at which the cell cycle continues or stops. External signals and growth factors can influence the cell cycle and affect the progress at or before this critical checkpoint. The G2M checkpoint allows cells that have successfully completed all three phases of interphase to begin mitosis. The last checkpoint is the spindle checkpoint, ensuring that all chromosomes have attached to the spindle in preparation for anaphase. Growth factors, the size of the cell, and the nutritional state of the cell are all contributing factors in cell cycle regulation, ensuring that only certain cells divide at appropriate times. Once all the checkpoints in interphase are cleared, mitosis can occur. From interphase to cytokinesis, the entire process of cell division can take on average 10 to 20 hours in a typical plant or animal cell. Depending on the nature and use of the cell, the process can happen at different frequencies as well. In humans, our skin cells have a high turnover rate due to wear and tear and go through mitosis very frequently, whereas other cells, such as adult neurons and muscle cells, rarely divide. The accuracy of mitosis, as well as the consistency of the checkpoints during interphase, ensure that most cells in a eukaryotic organism can produce identical copies of themselves. This process allows for growth and repair to prolong overall physiology as well as life itself.
Okay, that was a good video. I think it explains everything we just talked about. I'm going to put this on the blackboard, okay? Alright. There's a lot of videos there.